So it looks like a few others joined, so we'll go ahead and get started. Good afternoon, everyone. My name is Jeremy Stuhl, and I have the privilege of being your host today. And welcome to today's webinar, Creating Translationally Relevant Models for Immunotherapy Research, a closer look at humanized FcgR mice, presented by Dr. Zhiying Li. Dr. Li is a neuroscientist by training. Her PhD study focused on synaptic transmission and presynaptic vesicle recycling, which was conducted in the lab of Dr. Venkatesh N. Murthy at Harvard University. She did her postdoctoral training in Dr. Jeffrey Friedman's lab at Rockefeller University, where she studied cell type specific hypothalamic networks of metabolic regulation in the context of leptin signaling. Afterwards, Dr. Li joined Taconic Biosciences, focusing on genetically engineered rodent models for health and disease, supporting genetic science and compliance, as well as scientific and commercial production. She joined GemPharmatech US as Director of Life Science and Technology in October 2021. Feel free to ask questions during our webinar today using the Q&A button. Also, this is being recorded and will be accessible from the GemPharmatech website. Dr. Li, the floor is yours.
All right. Well, thanks, Jeremy. Thank you, everybody, for coming today. I'm actually quite excited to share this webinar presentation with you, representing my colleagues from China, who are actually the drive and the motion behind today's presentation. What I want to share with you, though, as you hear from Jeremy, this is overall a new field to me as well. So in the past year or so, I've been learning quite a bit from our team members from the US, from China. So today, I won't be able to promise you that I know everything and every detail, be able to answer every question you have. However, I can promise you that I'll bring all your questions. If I cannot answer them today, I'll bring it to our team in China and also experts in the States and get back to you. So when we get to that occasion, feel free to leave your contact, email, or any other ways so that we can get back to you after the fact. All right, so I'll get started to share my screen. I want to make sure everybody is seeing my screen and is presenting.
Yep, looks good.
Great, all right. So we're going to talk about humanized FcγR receptor animal today and its application for the immuno-oncology research and drug development. So this has been an effort in a while, not only by GemPharmatech, but also by other researchers in industry and academia. But recently, this poster, as I'm showing on the screen, really caught our attention, which is presented by Effimed on their AFM13 antibody as a therapy for refractory CD30 positive lymphoma. So what is new, impressive about this report and the study result is that this new antibody is able to achieve 100% response rate among these patients. And in particular, it was able to raise the complete response rate from 38% before to 62% among these patients, which is really a remarkable achievement in terms of this particular disease area. So looking at the mechanisms of this particular antibody, this has been reported previously, as you see from these two listed references. So if you can see my my arrow, my mouse. So this particular antibody is a four-headed or a tetravalent antibody. It's actually a bispecific antibody by design. On one side, two heads are recognizing the CD30 antigen on the tumor cell. The other side is a epitope, the Fab, that is recognizing the CD16A on the NK cells. So this is quite unique in the sense the CD16A is a FcγR receptor. In particular, is the 3A in the human population. So in this design, this bispecific antibody not only is engaging NK cell, it is engaging NK cell, particularly gaining onto the Fcγ receptor on the NK cells. So this, relatively speaking, this is a new way of how NK cells can be utilized to do immunotherapy and to suppress tumor growth. So as you can see from the bottom part of the slide, the mechanism in this particular bispecific binding event is that the immunosynapses is able to bring the tumor cell to the NK cell through this bipartite activity, and then the NK cell will be activated to release porphyrins as well as granzymes. These are the molecules that actually do the action, which is to kill the tumor. So going into a little bit more background to some extent, although everybody recognizes that tumor microenvironment has been a very hot topic in the recent couple of years, and at this year's AACR as well. So the graphic here is showing a typical tumor, which is confused, consists of a lot of tumor immune cells in addition to the tumor cells. However, with our research, people recognize this tumor environment, generally speaking, has been suppressive rather than promoting tumor suppression or tumor killing. What is contributing to this suppressive environment is a lot of factors, as you can see. So although we have NK cells, although we have T cells, P cells, these cells are not really suppressing or having a negative activity on the tumor cells, so that the tumor cells are able to escape from the tumor suppression, and in some cases actually lead to metastasis of the tumor. So there has been great studies from many different research groups showing, even though the tumor is growing, there are immune cells, both adaptive immune cells as well as innate immune cells. They are present in the tumor microenvironment, and in particular, they are actually serving a function to some extent. Given the, as an example, I'm showing you a particular study published in 2020. Here, the different immune cell types are characterized in quite a few different tumor types, which is on the top portion of the panel. As you can see from the color coding, although these cells are present, they are either serving as a positive prognosis, which means their presence actually promote tumor growth rather than suppressing the tumor growth. And then only the red dots, which annotates as a negative prognosis, that means their presence is actually inhibiting the tumor. For example, the Th1 or the Th2 cells in the pancreatic cancer incidence, as shown here. And then there are other incidents where cell types are showing in gray, there's no effect on the prognosis, where there's at the green dot shows they may have a mixed prognosis, even if in the same tumor type. So what is going on? And this has been a lot of effort to study how the cells can be, the immune cells can be reactivated to some extent, or re-engaged. So here is another way of showing what's going on in the tumor microenvironment. Showing here not only the immune cells, they are talking to each other. They're actually talking to the tumor in a way that just they're not killing the tumor as we wish. So all these interactions, they rely on signaling pathways, and in particular, as shown on the right side, these signaling pathways include cytokines and their receptors, checkpoint ligands and their receptors, as well as the classical MHC 1 and 2 to their receptor, the T cell receptor complex. So what is not, for example, here, very conveniently, between the T cell and a NK cell here, and a dendritic cell, the IL-15 and the IL-15 receptor are signaling the 4-1BB and the 4-1BB ligand, which is also known as the CD137, and their ligands are working between the two cell types. However, it's not leading to a tumor suppressive effect. In addition to all these, as the diagram shows, what is not shown in this particular diagram is that the Fc terminal portion of the antibodies, like these guys, they are actually talking to their receptors, which are called the Fc-gamma receptors. These Fc-gamma receptors are very prevalent on many different immune cell types. So this is what I'm going to show you in the next slide. Here on the upper right corner is a classical, antibody, right? It has the Fab region, which is the two little ears on the top, and then on the bottom, the Fc portion is our focus for today. So the Fc versus the Fab, the Fab. In this diagram, what has been known by people is that, or previous research, is that there are different types of Fc-gamma receptors. They are broadly categorized as activation, which is shown on the right side, versus inactive or inhibitory on the left side. I'll try to talk you through this diagram to give you a overview or a simple picture of how these Fc-gamma receptors are working in principle, as we learned. So for the activation of Fc-gamma receptors, they are present, for example, on phagocytes, PMNs, macrophages, or NK cells. When these receptors are activated, they can lead to phagocytosis. They can lead to binding or the enhanced antigen presentation by the macrophages. They can also lead to ADCC by the NK cells when their Fc-gamma receptor is activated. On the inhibitor side, these are particularly the Fc-gamma receptor 2 or 2B, as in the human situation. When they are activated, mostly these are Fc-gamma receptors present on the B cells. So it needs to be inhibitory because we don't want B cells in this instance to be super activated. Otherwise, it's going to kill the cell that is responding to these antibodies. At the same time, though, these soluble antibodies, when they are recognizing their antigen, which is the little squares here, they are capable to form something called a immune complex, which is basically a oligomer structure of the antibodies with their antigen, and then many of them come together to form that. So this particular complex, they're not really in the inhibitor pathway for the B cells, but their presence in some instances actually can engage even more of the Fc-gamma receptor activating cells, and then that can lead to an enhanced tumor killing effect, for example, in the context of the NK cell. So hopefully by this, I gave you a rough picture of the rough classification of Fc-gamma receptors, as well as how they function in the context of suppressing tumor growth. I want to give you a short 2 bullet points, which is we'll talk about quite a bit in today's sharing, which is Fc-gamma R3A, which is also called the CD16A. These are human nomenclature. It is a major activation Fc-gamma receptor, and is mostly responsible for ADCC and ADCP. And then in the reverse side, for the Fc-gamma receptor 2B, this is also in the human nomenclature. This molecule is also known as the CD32B. It can function as a cross-linker, which is basically to form the immune complex, which is not literally to inhibit the B cells from unnecessary overactivation, but is more, it can participate in the activation of the tumor killing cytotoxicity. So we'll talk about this a little bit more later today. So as a segue to demonstrate how this has been used in real clinical settings for treating human diseases, I'm showing you a MOA for rituximab, which is anti-CD20. This is a therapy for CD20 positive lymphoma or B-cell lymphoma. As you can see in the diagram, what happened is that the rituximab here, as illustrated, it has the red little dots recognizing the CD20 on the B cell or on the lymphoma cells. On the other hand, the Fc portion of this antibody in particular recognize the FcγR receptor. This is CD16A on the effector cell. In this case, is the NK cell. So when this pathway is activated and then very effective, this can lead to NK cell killing of the CD20 B-cell lymphoma. However, this has been really good, but there are also a population or subportion of human patients who has a variant of the CD16A. They are low affinity to the Fc portion of rituximab. So in these patients, this particular therapy, rituximab, has not been able to be effective or as effective in other human patients. So what drug development effort did is that they modified the Fc portion of rituximab to develop a new antibody, which is called the obinutuzumab. And this basically an enhancement of the Fc portion and is binding to the low affinity FcγR receptor 3A on the NK cell or the effector cell. Because of this enhanced binding and engagement of the effector cell, NK cell, for these patients, this antibody has been much more effective than the original rituximab in clinical settings. So this is all good, and there has been a very clever study to really demonstrate it is really the enhancement of the obinutuzumab Fc portion that engaged the effector cell and then enhanced ADCC effect on the tumor cell. So I'm going to show you some data from this study, and hopefully, I can explain to you very clearly. So basically, we have the two antibodies in our discussion, right? The rituximab is a human antibody. It does have the human IgG1, and Fc portion is human to recognize the FcγR 3A, which is also known as a CD16A on the effector cell. The obinutuzumab, though, it also has a human IgG1. However, its Fc terminal has been enhanced by glycosylation. So it's engineered to have glycosylation on the polypeptide, and that modification is going to enhance the engagement of the FcγR receptor 3A, and then leading to enhanced ADCC. So in order to have, so here, what is showing is these two antibodies, they have very similar binding affinity to mouse FcγR receptor. However, if you assay them against human FcγR receptor, you can see it is really the rituximab has a lower affinity to the 3A. The obinutuzumab has an enhanced binding affinity to the 3A. So this is the starting point. The engineering actually worked. So in order to have assay to look at tumor suppression, we have to have a mouse system as a model organism for the human, mimicking the human. So here, what was used are two mouse models, both of them having the RAG2 knockout, which is basically to enable the mouse to take in human tumors. And in one, so that's the control mice. In the experimental mice, these mice, their mouse FcγR receptor genes has been knocked out, and instead they have been given the human FcγR receptor as a transgene. So basically, both mice are immunodeficient so that they can be engrafted with human tumor. At the same time, the control mice is expressing mouse FcgR. The experimental mice is expressing human FcgR. As you can tell, here I'm showing you the IVIS, tumor measurements, right? So on the top is the control mice, and on the bottom is the experimental mice. So the control mice, they have mouse FcgR. They have been treated with untreated or vehicle control, or the isotype control, or rituximab, or the enhanced obinutuzumab. Similarly, the experimental mice has been treated with these 4 conditions as well. So in either one of the 2 mouse models, as you can tell, both antibody leads to significant tumor suppression, as you can tell by the fluorescence, right? On top of that, in the control mouse, both of the antibody treated group still have some tumor stayed with the mouse at day 28. In the humanized animal, that is a huge improvement compared to the mouse FcgR receptor situation. And in addition, as you can compare the lower panel, the rituximab versus the obinutuzumab, the humanized mouse with a Fc portion enhanced antibody really is literally giving a 0 tumor readout. So the results from the control mice versus the humanized mice are graphed in the in the 4 panels lower. So on the left side is the control mice. The yellow line is the obinutuzumab, and then the green line is the rituximab. As you can tell, the tumor, they both can suppress tumor compared to the the blue line. However, when you look at the humanized animal, panel 3, the difference is very obvious. And then the panel here, and then the last panel, these are survival rates among the animals. As you can tell, the humanized animal that is receiving the obinutuzumab treatment really enhanced the survival, which is obviously consistent with a much higher tumor suppression effect. So this result actually made us, the research team, really wonder what is going on here. Should we do a FcγR receptor humanized animal so that this animal can better support immunotherapy development, antibody efficacy and safety? So what we did is, to do some literature search in order to educate ourselves and have the proper knowledge when we jump into this question. So as you can see on the diagram, there is significant difference between the human FcγRs versus the mouse FcγR. To begin with, there are 3 subclasses among the human: 1, 2A, 2B, 2C, 3A, 3B. In the mouse, though, is only 1, 2B, 3, and 4. And as you can, in the middle parts, you probably wouldn't really be able to remember, but you can tell, different receptor subclasses have different affinity to the IgGs. And we'll have a bit more discussion on that. In the table, though, it is showing, in the mouse is relatively simple. There's 2B, which corresponds to the human 2B. However, the human 2A, 2C are absent from the mouse genome. Similarly, for the subclass 3, mouse has only 1, human has 2. And for the mouse, group 4, there's no human counterpart. Although I want to mention it to you here, though, the 4, although they are different, people still, our knowledge so far seems to suggest that the mouse 4 is equivalent to the human 3A. We'll have more discussion on that and see whether that's really a functionally relevant or important point. So here again is demonstrate the difference between the human and the mouse in a different way, which is by subcellular distribution. So as you can tell, human is on the top, mouse is on bottom, and human, there are like 7 different molecules, right? And then they are, quite differentially distributed among different immune cell types. On the mouse side, though, you can roughly tell the 2B, the 3, and the 2B and 3 in particular, kind of, a broad spectrum of subcellular distribution. 4 seem to have, monocytes or macrophages and neutrophils. And this is part of the reason why people think this is relevant, or the mouse equivalent to the human 3A, because human 3A and human 3B are kind of, cell type specific. And in the 3A situation is the macrophages that express the 3A. So that's the connection. However, we can think about whether, to think these are equivalents or functional equivalents, whether that's a good idea or not really a good idea. Different people may have different take on this. The other thing I want to, make sure that I mention to you, because we're going to revisit that later, which is the 3A, as I told you, is the CD16A. It is very specific for NK cells, which is relevant to ADCC and the macrophages. And then the 3B, although 3A, 3B are close relatives, the 3B has a different distribution. It's in neutrophils and some basophils. On the mouse side, though, you can tell that the receptor 3 class has a much broader distribution, and similarly, the 2B has a very broad distribution as well. And I mentioned to you, the class 3, and they are the activation type, and then class B is the, relatively speaking, the inhibitor type, or the type that can do cross-linking, forming these immune complexes. All right, the last slide to demonstrate differences. So we have been, using mouse as a model organism to test drugs, right? So here we are testing antibodies that are geared toward human antigen or human immune system, but we are using mice. We're providing a mouse environment for the testing. So it's quite critical to see how human antibodies and mouse antibodies are binding to the mouse FcgRs in the mouse environment. So on the left side is showing you the mouse versus the mouse FcgR. As you can tell, they engage the mouse FcgR quite well, in the sense they have high affinity binding to their FcgR receptors. On the other hand, the same mouse FcgR receptor may or may not bind to human antibody, as you see, in the case of IgG2. If that's a human origin, it has lower affinity to the gamma 2B or gamma 3 mouse receptor, and is binding to the R1, and R4 is basically absent. So by and large, hope I have give you enough information on. There's a good solid evidence that to evaluate human therapy in the mouse, and in particular, if the therapy is going to have a potential to engage the FcgR, we might think about how we can make the mouse a better system for the testing, and to do the translation from a mouse to the human. So that's really where we decided that we need to make a humanized FcgR receptor mouse. So on the engineering side, there could be different designs of how you can introduce the human genes, or how you can humanize the animal. Here is how we decided to do it. So first of all, the 4 classes of the mouse FcgR, they are actually located on 2 different chromosomes. So in order to get rid of them, is not a trivial effort. So we made knockout on either of the 2 chromosome locations, and we made a homozygous double mutant animal. This is for the mouse side. For the human side, it is probably even more challenging, because the entire human FcgR receptor, there are one chunk all living on chromosome 1. However, they span 40 megabase pair. This is not a trivial effort for any knockout or knock-in strategy. So what we did is to bypass the challenge, we took advantage of the back transgenic capability, which is we know the region of the coding sequences. We also know from a lot of back transgenic animals what's the flanking region, maybe the core pieces that is governing the translational or transcriptional regulation of the coding region. So we took quite a few back constructs carrying the human genomic DNA and fuse them together to make a 270 kilobase pair fusion back DNA, and then introduce this human back into the mouse genome as a back transgene. So basically, as I'm showing you here, we have the portion of the mouse KO and then the portion of the human back transgene in our humanized FcgR receptor mouse. I want to give you some words because we're gonna show you some data on the validation of the animal. So FcγR receptor 1 in human is also called CD64, and the receptor 2 also called the CD32, and receptor 3 is CD16. All right, having the mice is great. We have to make sure the mouse is engineered correctly and having the effective or the functional properties as we design the animal to be. So showing here is 3 sets of animals from the left to the right, wild type animals, and then in the middle is the animal carrying the human back transgene and a heterozygous mouse FcγR genotype. And on the right side is the humanized animal with a mouse knockout, a homozygous knockout on the mouse FcγR. So I'm showing you 2 cell types from spleen, the neutrophils and the monocytes. If you remember, there's a difference in terms of CD32 expression in the mouse versus the human. So the human and mouse monocytes, both of them express CD32. However, only the human neutrophil will express CD32. So in our validation, as you can tell, this is by flow cytometry. In the humanized animal, the neutrophils, they express CD32. Although in the wild type animal, they're not supposed to express mouse CD32, so they don't. And in the monocyte situation, the wild type animal express CD32 on the surface of the monocytes very well, as they're supposed to. And then in the fully humanized animal with the mouse knockout, is only the CD32 is only expressed in the human portion, as you can see. Next is CD16A. This is an important molecule for activating FcgRs, leading to ADCC, tumor cell killing. So in this panel, showing you the neutrophils and the NK cells. The neutrophil and the NK in the humans, they are very specific 2 cell types expressing the CD16A. In the mouse, it is not the case. The mouse neutrophil is supposed to express CD16A, but not NK cells. If you remember, we were talking about how functionally relevant it is between the mouse and the human. So as you can tell, in the neutrophils, yes, the wild type animal express the neutrophils express CD16A, and in the fully humanized animal, there's some expression here as well. Not really very much. When we move to the NK cell scenario, which is great, we can see there's a change from almost like no expression in the mouse to a good portion of NK cells in the fully humanized animal expressing the 16A. So you could say, okay, maybe here the neutrophils are not present. Our animal is not really, developing or producing neutrophils as they should. So we asked the same question. So what we did is we know in both humans and mouse, neutrophils are supposed to express the 16B, which is a 3B in the human situation. So we now, instead of using a 16A specific antibody, we use a different antibody which can recognize both 16A and 16B. So we use that antibody to see whether we can detect neutrophils and still be able to detect the NK cells. So in this situation, as you can tell on the top portion, it is recognizing the mouse neutrophils when it's using the mouse antibody, and it is also recognizing the neutrophils when it is a fully humanized animal with the absence of the mouse FcgR. The NK cell, again, is similar to before, this antibody could detect the NK cells that expressing the 16A. All right, so the last bit of validation, we also use flow, and this is, to look at the macrophages. Macrophages has been putting on a good momentum in the IO in the past year or so. And in this particular experiment, we do want to see, whether there's any macrophages that express CD16A. And if you remember, for the humans, the CD16A is very specific for the NK and the macrophages. So what we did here showing you is the wild type animal, the mouse, versus a animal that is actually heterozygous for the human FcgR. So it still has the mouse FcgR as a heterozygous. So in this experiment, in the wild type situation, the mouse the flow reports the presence of the macrophages from the peritoneal exudate macrophage extract. And in the heterozygous animal, both the mouse and the human population are present. So which is, basically telling us we believe our engineering worked. We have a fully functional animal, which is having the FcgRs, the human FcgRs expressed on the mouse immune cells, and then the cell surface presentation of the FcgRs are actually human rather than mouse. So moving forward a little bit, we did a in vitro ADCC. So what we did here is basically having a cell culture of MC38 tumor cells. These tumor cells are expressing PD-L1 because we want to use an anti-human PD-L1 antibody to elicit the ADCC effect. And then in the culture, what we did is we provided human PBMC as a positive control. We provided NK cells extracted from a B6 animal. So these are mouse NK cells. They're not supposed to be engaged by the antibody at all. And then we also extracted NK cells from our fully humanized animal, which don't have the mouse FcgR. So proof of principle, and we do know these, R bars is still too big, is a n equals 3 in each case in this experiment. But at least we think the NK cells that expressing the human FcgR receptor on the surface is being engaged by the human antibody and is able to lyse the MC38 tumor cells, which is good. One step further, which shown here is actually an in vivo tumor suppression assay or tumor killing assay, as measured by the change of tumor volume. So what we did is we have a collaborator who is developing drugs that is recognizing a particular tumor antigen on the MC38 surface. At the same time, these two antibodies are supposed to engage the FcγR gamma receptor 3 on the NK cell. So as you can tell, when these two, what they are different in their structure, how they can engage the Fc gamma receptor. So as you can tell on the right side of the panel, both of the antibody in comparison to human IgG, they are able to suppress tumor growth to some extent, not 100%, but at least as a proof of principle, it seems these two drugs are effective to some extent. And then from our perspective as a mouse provider, we think the animal is working. And then, for the same type of research, as I will share with you later, we definitely encourage people, and we are open to collaboration on fully characterize and use these animals for preclinical research and studies. So I've shown you there's some efficacy effects. We have done a preliminary experiment with one of our collaborators. When we develop drugs where I say them for efficacy, there's always the other side of the story, which is the adverse effects that these therapy may elicit. So here showing you is, any organ in the body pretty much are prone to be adversely affected by a particular therapy until we prove is safe. And using a particular therapy, a 4-1BB antibody, urelumab, as an example, as you can tell, this is a publication in 2017. As the doses of this particular antibody increase from 0.1 to 0.3 to 1 milligram per kg, the adverse effect and the intensity, the broadness of the adverse effect actually increases. And then, from level 1 until level 3. And then the x-axis is the dose of the drug. So this particular study really made people think, what's going on with this this clinical result, because everything looked very well when it was preclinical evaluation. So here, I think is, in my opinion, it will become a very classical experiment, which is actually looking at how the 4-1BB antibody was working in the pre and clinical phases, and where is the fundamental contributing factor between the difference of the preclinical results versus the clinical results. So is a very complex paper. I want to take you through, and I hope I can provide you enough information as well as clear description of the data to walk you through. So in this particular experiment, the group actually designed two 4-1BB antibody, the 3H3, which is a high affinity to the CD137, and a LOB 12.3, which is a low affinity to the antigen. However, at the same time, they gave these two antibody, or these two Fab region, a FC region, two different FCs. For the high affinity Fab, they gave a FC region, which is to engage the activation FcgRs. And then for the low affinity FAB, they gave a FC which can engage the Fcγ2b, which is the inhibitor or the cross-linking FcγRs. So as you can tell, they used two tumor cells to look at efficacy of these two antibodies. So the CD26 or the B16 ova tumor, in both situations, these two drugs, whether the Fab is high affinity or low affinity, they can kill the tumor very effectively. When they look at the adverse effect though, so in the bulb animal, which is correspond to the CD26, and then the B6 is corresponding to the B16 ova tumor cells, the antibody itself, especially the one with a high affinity, elicits a strong adverse effect as shown by the liver toxicity, the ALT measurement. As you can tell, the low affinity one has a much better adverse effect profile. At the same time, they also show just use a wild-type BAB or a wild-type B6, the high affinity antibody can still elicit a liver toxicity effect. So which shows the Fab region is actually irrelevant in terms of eliciting the adverse effect. Is basically the Fc region that is giving rise to the adverse effect. Now looking into this matter, how is this Fc region, different subsets of FcgRs are engaged by the two antibodies. How is that leading to the differences between the two antibodies and the effects they have seen? So they took a reduced system, which is a cell culture in vitro. They have the T cells in the culture or co-culture with another cell. The other cell is transfected with different subtypes of FcgRs, so that when they provide an antibody, the binding of the antibody to the FcgR on the co-cultured cell, whether they will look at whether that binding and also the binding of the antibody to the C137 on the T cell, whether this chain of reaction can lead to interferon gamma receptor, interferon gamma release, which is a readout of overactivation of T cell, the adverse effect of T cell activation. So as you can tell, when they are supplying different FcgR subtypes in the co-culture, pretty much whatever the subtype they provide, that can enable the T cell to express interferon gamma. And in particular, the FcγRIIb expressing situation actually elicited the highest amount of interferon gamma receptor interferon gamma release, which is to some extent surprising, because we're thinking probably the activation type of FcγRs, which is on the high affinity Fab, that should give the highest amount of adverse effect, right? However, this also making sense because the FcγRIIb is capable to do the cross-linking, which in the by and large will be able to enhance even more of the activation of the FcγR response. So they took another step further. Actually, instead of using a reduced culture system, they use different types of knockout animal for the FcγR. And in this particular case, as I highlighted in the red rectangle, so when they test the two antibodies, right, high affinity, low affinity, with a activation FcγR versus a inhibitory or cross-linking FcγR, in this, when the FcγRIIb is knocked out, the high affinity efficacy is not affected, meaning high affinity itself from the Fab is enough to convey the tumor suppression effect. However, the low affinity antibodies are completely affected by this effect, meaning without the cross-linking effect, this low affinity antibody is not able to kill the tumor. And then in the lower part of the graph, as you can tell, what they did is a very smart design. They give the high affinity Fab with a FcγRIIb engaging Fc region. So having this, although the Fab is still effective in killing the tumor, we are still seeing a good effect. However, that effect is attenuated compared to when the Fc gamma region is the activation part. So this particular experiments tells us both the high affinity and the low affinity region actually rely on, or to some extent, take advantage of the Fc region and their activity of with the Fc gamma receptor on the NK cell to fully exhibit their tumor suppression activity. And then for the rest part of the experiments, as is shown, to get rid of the FcγR3 or the FcγR1 is not really affecting the efficacy of the antibody, because when you knock out one subclass, there's the other activation subclass still present. And then the last bit of data I think is very interesting is the use of wild type animal in their hands. So what they did is a very clever way. Not only they can switch the Fc region of the antibody. So this is the low affinity antibody. This is the high affinity antibody. They give them, the activation part, or the part that can engage the cross-linking, or they silence the Fc region completely. So the antibody can only rely on the Fab portion for its effect. So as you can tell, in the low affinity antibody situation, only the part which has the Fc portion that is able to bind or able to elicit the cross-linking, that can lead to effective tumor killing. Otherwise, either the silencing as the green, or having the Fc portion that is activating the activational FcgR is not going to lead to a tumor suppression effect of the low affinity antibody. On the high affinity portion, as you can tell, both of the one that is engaging the activation or being silenced, they can suppress tumor growth. This tells us the high affinity antibody doesn't really need the Fc portion to do the job. However, when you give them the Fc portion to do the cross-linking, it actually failed the tumor suppression. And then this is the last bit of their experiment. I think, I would point out to you, bring your attention to these, the two on the left side, which is, switching out the activation or the activational FcgRs. In either case, the high affinity antibody is still able to elicit a toxic effect that is read out by the ALT activity. On the other hand, when the FcgR2b is knocked out from the animal, neither of the high affinity, the high affinity antibody is not able to elicit adverse effect anymore. So this tells us, for both of them, the inhibitory pathway versus the activational pathway, both of them actually contributed to the efficacy and the safety profile of the high affinity and the low affinity antibody. So as a summary in the paper, you can check if you like. The researchers are proposing a way of how we can design antibodies, not only based on their affinity at the Fab region to their antigen or the tumor antigen, but also rely on the Fc portion, how the Fc can be engineered to be, recognizing different pathways of the FcγR receptors, as well as how that tuning, the binding efficiency or affinity can be tuned by doing antibody engineering. So if you like, you can take a detail in the paper. So here is a set of experiments that we did in collaboration with Crown Bio. We were thinking, all right, that's a great experiment to demonstrate how the 4-1BB antibody showed what it showed in the preclinical versus the clinical. So we were wondering, where is the 4-1BB urelumab is in terms of the Fab affinity versus the Fc-gamma receptor engagement. So where is that antibody in these two aspects? So what we designed is, here is the urelumab by itself at two different doses, shown by orange and red. As you can tell, they can suppress tumor. We're using a Bob and humanized animal with the Bob tumor to do this experiment. What we also did is we give the urelumab, which is human antibody, a mouse tail, a mouse Fc in this particular situation. So we were thinking, okay, if this antibody is already, activating the activational FcγR, probably this switch would not do very much. However, to our surprise, this actually further increased the efficacy of this antibody. And then you can tell this, the body weight for all the different groups are pretty much similar. So we think probably we have a readout, which is if, let's say, retrospectively, if the urelumab was given a mouse tail, a mouse FC, which can engage the mouse FcgRs, probably at the preclinical phase, the adverse effect would be read out by using mouse, for example. As you can tell, by 2 days or by 7 days, the ALT activity is very obvious, especially the 7 days. And then, if we have the engineered antibody, which is, the anti-mouse 137 as a surrogate to the 3H3, even in the wild type animal, they are able to show a obvious liver toxic effect. And finally, this is just to show the adverse effect as shown by the ALT. This is by morphology, the liver tissue morphology. All right, so I hope I have provided you a good set of information of how we rationalized there's a need for us as a mouse provider to develop a human FcgR receptor animal for the efficacy and the safety testing of next generation IO therapy or candidate drugs. And I have provided you enough information of how we engineered our humanized FcgR receptor animal. And these animals has been in the cooking for a bit. So we are now very happy to share with you, they're ready for sale, and we are actually accepting orders. We expect the production scale to go up to 100 female animals in July. So remember, this is not a trivial effort. These animals have double mutant or double knockouts on the mouse side and a back transgene on the human side. And as I said before, no matter how much we have been, characterizing these animals or validating these animals, what we can do cannot cover everybody's needs. So we are still open to collaborations to, further characterize these animals for any particular or specific use that our customer may have. So with that, I have one last slide to share with you, which is we do think this animal could play a very important role in further validate combinational therapies or combinatorial therapeutic developments in the near future. So we have been putting an effort to develop more complex or compound humanized mutant animals. So as you can see from this slide, we are crossbreeding this animal with quite a few other humanized immune checkpoint animals that we already have in our pipeline. And then at the very last, we're considering to develop a immune deficient animal that is expressing the human FcgR for some other therapeutic needs. So with that, and well, as the dots, the different coloration give you a sense of where we are with the pipeline, whether we already have F0, F1, or the project is just to be started or already started, still at the early stages. So if you're interested, you're more than welcome to contact us and then start discussing with us. So with that, I'm going to close for the presentation from my side, and I'll be open up for some questions or discussions.
Yeah, thanks, Zhiying. That was a great presentation. Excuse me. We are just at the hour mark, so we don't have a whole lot of time for questions, but we do have 2 questions that I hope we can get through relatively quickly. The first question is there genetic evidence linking FcγR biology to immune cell activation?
I think there is, I don't know whether, this, the participant who came in late that missed the part. I think, some of the references I cited actually showed very good evidence, in my opinion, that the FcgRs actually can play an active role in the effectiveness of the IO therapy. In particular, if you remember the rituximab versus the obinutuzumab evaluation, that paper. So if you want, I can put the reference in the chat, or share with you after the fact if you email us. All right.
And there's actually two more questions. One just came in. I think the second one here is related to the first. What about having human neonatal Fc receptor there as well?
Okay. I have to say, this is a question I'll be more than happy to bring back to the R&D team. What I know is we do have a FcRn humanized animal in our pipeline already. I think there has been a discussion to bring these two humanized animals together to form a more complete humanized animal using this. So for this participant, if you are thinking a particular direction for such a, the neonatal FcgR or the entire FcgR family of proteins, more than happy that we have the discussion with you.
And then a follow-up is, what alleles are used in the human FcγR mice?
The humans, we basically give the mouse the 1A, 1B, 2A, 2B, 2C, 3A, 3B, everybody.
And then finally, are the hFcgR mice are in immunocompetent B6 background?
Yeah, right now the one that we scaled up the production, that's on the B6 background. They are fully immune competent, and as I showed on the last slide, we are developing a PRKDC knockout animal, and then giving these animals the FcγR, the human FcγR. So this animal, if we make it, that's going to be on an immune deficient background. So you're more than happy to talk with us. That's in our pipeline, and we agree with you. There's a value for these animals for IO studies.
And one final question just popped up here. Is the CD16A allele the high affinity or low affinity receptor?
No, since I have that slide, I'm not gonna lie. I'm gonna go back and check it for you. So the human 3A is a high and low affinity. So I guess depending on the antibody, depending on the IgG type, the affinity may be different. Oh, I have the reference if the this participant want to know. I can copy and paste in the chat box. Right. I hope that helps.
Looks like it just went to the host and panelists, so I'll choose.
Oh,
so
sorry.
You can see it.
So as I do that, I'm going to also post the other reference in the chat box, which is the rituximab versus the obinutuzumab study. I think is a great study, is a great after the fact revelation for every at least I learned a lot from that design. Learning from the human, going back to the mouse, using the mouse as a way to really tease out the molecular mechanisms. I think that's a great study.
So that looks like all of our questions. We have gone over our time. So I think that we'll just wrap things up for this month's webinar.
Sure. Well,
okay.
Yeah, I took a little bit more than I wanted, and I thank everybody for staying with us, and for the questions. Always happy to discuss, and have a good day.
All right, take care, everyone. See you next month.
All right. Thank you, Jeremy.
Thank you.
Yeah, see you in a bit.